Hi, I am Maggie Zhao, Metware Product Manager. Today, I will continue to explain spatially resolved metabolomics to you. In the last two videos, we started with what is spatially resolved metabolomics. That means spatially resolved metabolomics can allow you to do in situ analysis. And then we introduced why we choose spatially resolved metabolomics. That means the specific application direction of the spatially resolved metabolomics. In this video we will share with you how to do spatially resolved metabolomics. First, let me introduce the experimental principles and technical methods. So far, there are four main technologies for spatially resolved metabolomics, and MALDI is the most widely used one. The experimental principles of MALDI are relatively simple. If you want to see how it works, you can watch our first video, What is Spatially Resolved Metabolomics? After the initial understanding of spatially resolved metabolomics, let's take a look at the specific experimental procedure of it. First, you need to get biological samples. Then, the samples are supposed to get a preliminary treatment sectioning. Second, we spray substrate on conductive glass after slice processing. So here's the question, what's the use of spraying matrix? It is mainly used to coat the metabolites on the surface of the samples. Now comes to the third step. The third step requires laser irradiation on the slide with the sample, so that the metabolites into the mass spectrum detection. A laser is a beam of light, and the size of the beam causes the size of each beam. Thus, the spatial resolution of the whole sample is different. When a certain size of laser beam shines on a sample, the matrix on the surface of the sample was co-crystallized and sublimated with metabolites, which were then detected by mass spectrometry. Finally, the qualitative and quantitative information of metabolites was obtained. How to get information about its distribution? We notice that on the slide, our laser beam is of a certain size and illuminates the slide in turn. Every point it illuminates has a position information. Therefore, we can in situ image the spatial distribution of metabolites in different locations of samples. So those are the four main steps of our MALDI experiment. Next, we will explain the four steps specifically. First of all, we need to prepare reagents and consumables before sampling, such as sterile scalpels, scissors, and tinfoil. After preparation, we need to do sampling. Second, we can cut off the relative part of animals and plants, then rinse the contaminations such as bloodstain and pelage with prepared stroke physiological saline solution. Quick freezing the clean samples in liquid nitrogen. Third, place the liquid nitrogen in a foam box and fold the aluminum foil into a small boat, floating on liquid nitrogen. Place the sample in the tinfoil until its surface turns to completely white. Then put the tinfoil into freezing tubes. Fourth, storage and transportation. You'd better store the processed samples at -80 degrees. Dry ice is generally recommended for transportation. Now, we have understood the sampling process, let's take a look at sample pre-treatment. Some samples we can do spatially resolved metabolomics detection directly, like fresh samples. In addition, some samples need to be embedded. What kind of fresh samples can be tested directly? Samples with fewer cavities and lower water content, such as routine tissues, brain, liver, kidney, etc. However, for samples with relatively high water content and many cavities such as eyes and plant seeds, embedding is required to ensure the integrity of slices. How to choose embedding agent? It is generally recommended to use CMC or gelatin for embedding. However, we do not recommend using OCT or paraffin for embedding, because it will affect the final results of our mass spectrometric imaging detection. After preliminary processing, the next step is to conduct the experiment. Before the experiment, we need to choose the resolution. How to choose the resolution? There are three principles. 1. If you do not need to pay attention to microstructures, we generally recommend to choose 50 micrometers because it is cost-effective and the price is not much lower than 100 micrometers. In addition, the high resolution of 50 micrometers can meet most of your requirements and can get relatively clear mass spectrometric imaging results. However, if your samples are special, too small, we need to ensure that the resolution is greater than 1000 pixels. Only in this way, its experimental results may meet the needs of the overall research. For example, in this root sample, the root diameter is only 800 micrometers, and when you select a resolution of 100 μm for the entire sample, its total number of pixels is less than 64. Results obtained may be chaotic, and the data of spatially resolved metabolomics cannot divide the samples into regions. Therefore, it does not meet our experimental needs. 2. When your samples are regular samples and you need to pay attention to microstructures, the resolution size is required to be smaller than the size of the microstructure. For example, the conventional diameter of a rapeseed is between 1.27 and 2.05 millimeters. The size is suitable for 100 micrometers resolution. However, the seed coat is only 26 to 28 microns in size. So, obviously, we can't apart the seed coats by using 100 micrometers resolution. Therefore, 5 micrometers is recommended for the later experiments. After choosing the resolution, the mass spectrometry experiment can be carried out. Next, let's take a look at experimental maps. The distribution experimental spectrum of brain's metabolites were shown here. The first is a 150 micrometers image resolution, which shows that metabolites are distributed very regularly in different areas of the mouse brain. Next, 50 micrometers image resolution, and you can see that the 50 micrometers resolution map is much clearer and much denser. And then we have 10 micrometers. It is basically showing the upper right corner of the 150 micrometers. As you can see, 150 micrometers only roughly distinguished the structures. When we do experiments with 10 micrometers, it can specifically divide the brain into regions, and the metabolites distribution of each regions can be displayed much clearer. This is the difference between high resolution and low resolution. If you still have questions about what is spatially resolved metabolomics or why choose spatially resolved metabolomics, you can contact us. Our sales will introduce our MSI products to you. That's all. Thank you for your watching.